Hello, my name is Sawyer Hancock, and I will be presenting on how multi-pesticide exposure at the Apis mellifera colony level during royal jelly production negatively impacts queen bee development, survival, and reproductive health. Apis mellifera greatly impacts not only the environment, but also the world's economy, as honeybees represent the majority of insect pollinators. Honeybees pollinate a wide variety of plants such as fruits, vegetables, nuts, and field crops. While doing this, they help maintain our ecosystem. The decline of Apis mellifera threatens agricultural productivity, as one-third of the world's food is derived from plants that benefit from pollination. The table I have included on this slide outlines crops that are dependent on bee pollination. The most important honeybee in any hive is the queen bee. The queen bee differentiates from all other bees by many factors, the most important being that they are responsible for all reproduction in a colony. Queen bees start off as larvae, just like all other bees. But what makes a queen bee so special is the diet that they are fed within queen cells. This diet consists of royal jelly, as opposed to the normal diet of honey and pollen. This diet of royal jelly is essential to queen bees as it helps develop their reproductive capabilities. There are a few common pesticides that have the potential to interfere with the nutritional components of royal jelly, therefore indirectly affecting the queen bee's overall development, survival, and reproductive health, since royal jelly is the basis of what makes a queen bee a queen. Without a healthy queen bee, colonies will eventually collapse, so the integrity of royal jelly's nutritional composition is extremely important. This leads me to my overall question of what impact does multi-pesticide exposure during the royal jelly production process have on the queen bee's development, survival, and reproductive health? In my research, I have found that exposure can fundamentally alter the composition of the royal jelly as it influences phytosterols, amino acids, and protein profiles. This can lead to poor reproductive development and stress of an up-and-coming queen bee, which impacts the colony. In the next four slides, I will explain the research that was conducted that resulted in these findings. A simulation of multi-pesticide contamination and pollen during Apis mellifera royal jelly production was conducted by researchers. This simulation separated six naturally made colonies, three being control groups and three being treated groups. The treated groups were fed pollen that was mixed with 9 different pesticides as shown in the attached chart. The pesticides were diluted with acetone prior to mixing the pollen. The control group was fed pollen with an equal volume of acetone. Consumption was recorded for 46 days, as this allowed ample time for 2 brood cycles to ensure nurse bees uh, were reared while exposed to pollen treatment. After the conclusion of 46 days, each colony was moved to swarm boxes. During grafting, larvae were removed, as well as queen cells. Each queen cell has its own royal jelly removed for testing. Phytosterols were among one of the first components to be tested. They are a micronutrient that is essential for the production of hormones responsible for insect molting and integrity of the cellular membrane. In this test, 7 phytosterols were analyzed. The results shown in the graph, green being treated and red being controlled, reflected that there was a significant lower amount of phytosterols in treated groups versus controlled groups, including an important phytosterol, 24-methylene cholesterol. 24-methylene cholesterol is an important dietary component for brood rearing. Additionally, amino acid concentrations differed between controlled and treated groups, which may be indicative on implications to larval food nutrition. The most important difference was that 10-HDA was found much higher in controlled samples. This biomolecule is an antimicrobial and is considered to be a freshness marker. Lastly, protein profiles significantly varied between the controlled and treated groups. Important proteins such as MRJP1 and glucose oxidase were found to be lower in treated groups, while defensin 1 was found to be higher. MRJP1 is an important component for rapid development and reproductive maturation, and, and glucose oxidase helps aid in the prevention of microbial infections. Furthermore, the increase of defensin 1 suggests increased stress in nurse bees. Both glucose oxidase and defensin 1 are crucial for food sterility. Overall, multi-pesticide exposure can have a great effect on the nutritional components of royal jelly, which indirectly affects the queen bee development, survival, and reproduction. Without proper nutrition, queen bees are unable to develop physically and reproductively the way that they are supposed to. If queen bees are not healthy, entire colonies could suffer. The overall significance of this research shows us how multi-pesticide exposure during royal jelly production at the Apis mellifera colony level will affect queen bee's health. At this time, long-term effects are not known. I believe it is safe to say that without healthy queens, we could potentially see bee colonies decline. Declination of the Apis mellifera would severely impact not only the environment but also the economy as well, since a larger portion of global crop production is dependent on pollination. This concludes my research over how multi-level pesticide exposure at the Apis mellifera colony level during royal jelly production negatively impacts queen bee development, survival, and reproductive health. Thank you.